this picture presents how, in general, the drug development process look like. And the process consists of three main stages. The first stage is discovery, followed by preclinical research. It ends with tests on animals. The second big stage are clinical trials. This is basically tests on humans. And the last stage is just the review process by Food and Drug Administration. And what is worth mentioning is that the whole development process is very long. It takes on average about 12 years and is extremely expensive. The average cost is about $2.5 billion. So I think that this is a good motivation to look for a method that can speed up this process and, of course, reduce costs. And basically, right now where most of machine learning research is being done is only this first stage, so the stage of drug discovery. And to understand the basics of the drug discovery, you should know two things. What is a target and what is a lead? So target is this white guy here. So this is basically, usually this is a protein that is somehow involved in a disease process. And a lead is just a drug candidate. So this is this green guy in this blue pocket. And what we want to do, we want to find a drug candidate that will bind with this target and reduce the activity of the protein target, and so slow down the disease process or stop the disease process at all. And there is a huge progress in applying machine learning both to target identification and validation and to lead design and optimization. Maybe you have heard about AlphaFold system. It is a system released 2 weeks ago by DeepMind, and actually the system is able to predict a 3-dimensional protein structure based on a sequence of amino acids, and is doing it really accurately. But during this talk, I will focus only on the second part, so on lead design and optimization. By leads, we understand drug candidates. Okay, so just to give you a brief introduction how deep learning can be applied to lead design, let's imagine that on the one side you have a molecule, and on the other side you have some properties and, for example, activity against a specific target. So what you can do, you can apply machine learning to do predictive modeling. So taking molecule as the input, you would like to predict some properties that are measured, normally are measured in the lab, or to predict the activity against a specific target. And there are several applications. Just to show you some examples, for example, we can predict activity. So this is just a classification if a specific molecule is active against a specific target or not. We can predict ligand-protein interaction. This is more or less the same, but as the input, we take molecule and the target simultaneously. We can also predict binding affinity. This is also similar to this, but this is regression problem. These two are classification problems. We can also do something that is called ADMET prediction. This shortcut ADMET stands for absorption, distribution, metabolism, excretion, and toxicity. So these are basic properties of the molecule. And of course, we can think about the inverse process. So we can consider some properties that we would like to have, and then we can try to model this mapping to generate molecules that possess such properties. And in this group, we have something that's called de novo generation. This is basically just generating new molecules. And we also have something like lead optimization. This is just optimizing the existing molecule, for example, to improve some properties. And of course, we are all in the field of machine learning, so there is no machine learning without the data. And if you're interested in doing some research in this area, there are quite many publicly available data sets to solve different problems in this group. But I would like to mention one thing, because these data sets are not nice and elegant data set like, for example, ImageNet in computer vision. There are many different kind of problems with them, and these problems include high internal correlations between example, bias, different kind of noise, and many, many errors. And so basically, if you are trying to use the standard methodology that you are like dividing your data set into training, validation, and test part, your error estimation might be very, very over-optimistic then. So be careful on doing that. Okay, the next thing we need to consider if we're thinking of doing deep learning for drug design is how we will represent our molecule. And there are several ways to do it. So the most popular way how we can represent a molecule is called smiles. This is basically representing the molecule as a sequence of characters. And I think I don't need to convince you that this is the most popular representation that machine learning community is using. Basically because you can easily adapt models that are being used in the NLP applications to that problem, so to process just a sequence of characters. The next representation is graph-based representation. So you represent atoms as the nodes, and you represent bonds as graph edges. And during the rest of my talk, I would like to focus on this representation. And the last one is three-dimensional representation. So basically, you just represent your molecule as a point, as a cloud of points in three-dimensional space. Each of these representations has pros and cons, but maybe I will leave it for further discussion. Okay, on this slide, I would like to show you how you can think about doing research in drug discovery. By doing research, I mean, of course, doing deep learning research. So at the beginning, you need to select one or maybe more tasks that you would like to solve. And you must decide on a specific representation that you'll be using. Then, of course, it depends what kind of task are you selected. So you also need to choose supervised or unsupervised framework, and then design the model architecture. So for example, if you decide on generative adversarial networks, you need to decide what kind of discriminator you will use and what kind of generator you will use. And for example, if you take such combination, so the task is ligand-protein interaction, 3D representation, supervised learning, and three-dimensional convolutional neural networks. This is actually a paper by Wallach et al. From 2015. If you take such representation, such combination, so activity prediction, smiles, CNN, and RNN. This is paper by Staszek Jastrzębski, who is the next speaker here. Hi. Okay, taking ligand-protein interaction, graph representation supervised, and graph convolution networks. This is actually a paper by me and my colleagues from 2016. And you can do similar game for de novo generation, so for unsupervised learning. So for example, taking smiles, variational autoencoder, and recurrent neural network. This is actually the very first paper in this topic by Gomez-Bombarelli from 2016. This combination, so convolutional neural network and differentiable neural computer as the generator, is actually the paper from this year, and this is, best to my knowledge, some kind of state-of-the-art in this problem. And there is also very nice paper that is using graph representations, also GAN plus reinforcement learning, MOP, and graph convolution neural network by Nicola Dicao and Thomas Kipp from this year. And I would like to explain myself a bit. What you can see on the right, this is not any kind of comprehensive literature review. These are just a few examples how you can do research here. If you're interested in this topic, just send me an email and I'll send you back a more complete list of papers. And of course, there are also other combinations, like for example, taking 3D representation with any of unsupervised learning framework. And these actually are still open problems. So you can think of doing some research in this area. All right. And in the rest of my talk, I will focus on these two papers. This, I know it, and this, I like it. Yeah, but seriously, they have two things in common. They are using graph-based representation and graph convolutional neural networks, and they cover two different areas of application. One is predictive modeling, and the other one is generative modeling. All right, so let's move to the first area of the applications. So to demystify what are these mysterious graph convolutions, let's begin with the ordinary convolutions on images. Probably you are all familiar with them. So let's imagine that we have a convolutional filter. This is actually a set of convolutional filters. And we have also an image. And we would like to take the inner product of this set of convolutional filters with this particular patch of the image. So let's highlight the central pixel in this patch. So you can think about this way, that you have a pixel here, and you have a set of weights that is actually associated with this pixel. So during the convolution, these weights will be multiplied by the value of this pixel. Of course, this pixel has this set of weights associated with them, and so on. And now let's take the graph, and also highlight some central node in the graph. You can do the same. You can associate a set of weights with each of neighboring nodes, and of course with the central node. So this is pretty similar, but there is one difference. This graph convolution, the convolution on the images, the neighborhood of the central pixel is well structured and is always the same. However, in graph case, the number of the nodes, the number of neighboring nodes may differ for different nodes. So also there is a clear way how to do ordering of the nodes. So how to solve it? One way to solve it is just to do the weight sharing between those filters, because then you don't need to worry what is the proper ordering. And just to dive into a math a bit. So this is just a convolution. This is just one layer of graph convolutional neural network. So basically what you are doing, you are taking features from the ELF layer of the central node and multiply them by the weights associated with the central node, and you are summing it with features of the neighboring nodes multiplied by the weights associated with the neighboring nodes. And there is also a kind of normalization constant here. This is because the number of neighboring nodes may differ. So to keep the signal level more or less the same, you need to normalize it. And of course, if you are stacking the consecutive layers, what you are doing, you are actually learning more and more abstract representation for each node just by gathering the information from wider and wider neighborhood. And graph convolutions have many extensions. For example, support for edges. You can add attention mechanism. You can set this normalization constant fixed, or you can make it learnable. Finally, you can easily implement this graph convolution using node feature matrix and adjacency matrix. Just to explain you maybe what it is, node features are, let's imagine that we have 3 types of atoms. This is carbon, this is nitrogen, and this is oxygen. So you are just using one hot encoding. So for example, this is 1, 0, 0, and it's carbon. For example, this is 0, 0, 1, and this is just nitrogen. And adjacency matrix is just representing the connections between the atoms. So for example, if the first node is only connected to the second node, then you have in the first row 0, 1, 0, 0, 0. I think that's easy. Okay, and to give you the example how graph convolutions can be used to predict some properties of the molecules. So in this paper, we proposed the following architecture where we have 2 separate deep neural networks. One is taking a small molecule, a drug candidate, as the input, and the second one is taking a fragment of a protein, basically the binding pocket, so the place where the small molecule is binding to that protein. And first we use a couple of graph convolutional layers to proceed these two objects. Then we are using a kind of pooling operation, so we are doing just softmax plus some pooling to basically to pool the information from all atoms into one representation vector. Then we are processing it using fully connected layers. And at the end, we are just taking a kind of similarity measure. So basically, it can be just the inner product between the 2 latent representations at the output, or you can take some kind of cosine similarity or whatever. There is a nice analogy of this model. So if you are familiar with models, deep learning models for information retrieval, where you can take a query and a document as 2 inputs to the deep learning model, and then you are just predicting if query fits to the document. So this is more or less the same. So you can think of a small molecule as a document, and about this pocket as a query. All right, so we were using 2 types of graph convolutions, and using one of them we achieved quite nice results. Then we compared it with 2 well-known methods for this ligand-protein interaction prediction. I will also did a kind of error analysis, but maybe I'll skip it for now. Okay, and now let's move to the generative modeling. So just to give you the idea what the generative modeling is all about. So let's imagine that you have your representation space. So in our case, this is just a space of all possible graphs. And of course, not every graph is a proper molecule. I would say more. You have to be really lucky to find a point in this space that will be actually the valid molecule. So basically, valid molecules actually lie in a kind of subspace in this molecular space. So, and basically what we want to do, we want to just find the mapping between this subspace, this molecular space, and this representation space, and we'll be doing it using deep generative models. Maybe just to clarify one thing, because these two pictures suggest that this is a continuous problem. Actually, it isn't, because graph space as well as molecular space, these are discrete spaces. So this is just a schema. Okay, so just to give you some information, so molecular space is really huge. It is estimated that it varies from 10 to the power of 50 up to the 10 to the power of 100. And also there are several properties that you would expect that this kind of generative models will have. So you would like the generated molecules to be valid, unique, novel, and diverse. I think that these properties are rather universal for generative models because, for example, if you're using your generative model to generate pictures of dogs or cats or whatever, you also want the pictures to be valid, you want them to be unique, novel, and diverse. But also, there are many properties that are specific to this particular problem. So for example, you would like the generated molecules to be drug-like, so they are similar to true drugs. You want them to be synthesizable, so basically that you can synthesize them using known chemical reactions, that they will be soluble, and many, many others. And basically, if you consider doing generative modeling, there are two leading approaches that you can use. One are variational autoencoders, and the second one are generative adversarial networks. And if you are thinking about applying variational autoencoders to graphs, there is one problem with this. So if you look into the loss function of the variational autoencoder, you have this factor, which is basically the reconstruction error between the output and the input. And if you are representing your graph as two matrices, so as node feature matrix and adjacency matrix, if you permutate the rows of those two matrices, this remains the same graph. So you can't consider that you will have the same ordering in the input and in the output. So your loss function must be invariant to permutations of the rows in those two matrices. And basically, this requires expensive matching function, for example, Hungarian algorithm. And this is a quite huge limitations of these methods. So there is a nice paper by Simonovsky and Komodakis from this year, which actually where they are actually presenting this kind of variational autoencoders for graphs, but their solution is limited only to the small graphs because of the computational problems. There is also no clear way how to linearize graphs, so how to present them just as a sequences. So maybe we should decide on the other approach. So maybe we should decide on the generative adversarial networks, because these models don't have such problems with the reconstruction measure. The only drawback they have is they don't have inference model, but we have to live with that. Okay, and there is really nice paper from this year by Nicola Dicao and Thomas Kipf, where they are actually proposing such GAN model for generating graph-based molecules. And what they are basically doing, they are using just multi-layer perceptron for generating adjacency matrices and node feature matrices. The generations are in continuous representation, so then they are doing just sampling. Sampling, of course, is a non-differentiable operation, so they are just applying here reparametrization trick with Gumbel softmax. And they are using graph convolutional neural networks as discriminator and also as a reward network, because they are combining actually generative adversarial network with reinforcement learning. They are doing it because they want the generated molecules to also have high value of quantity that is called quantitative estimation of drug likeness. So they want to force the model to produce molecules that actually are similar to true drugs. And the generator is trained jointly via GAN and reinforcement learning objective. That objective is just a combination of those two. There is also one problem with that model. It suffers from mode collapsing. This is a typical problem for GANs. And when they are presenting the results, you can see that the uniqueness of the generated molecules is very low because of that problem. Here we can see some examples. So these are true samples from QM9 dataset. And these are the examples generated by the MolGAN model. So they look pretty nice. And finally, at the end, I would like to tell you about one more technique. So let's imagine that we have already learned a kind of generative model, so a mapping between this molecular space and this representation space. And now we have some kind of function over the molecules. And for example, this can be like a combination of this drug likeness, and this is, I don't remember the name, but this is basically how easily you can synthesize your molecule. This is just the function that can estimate that. And of course, you know some values of the function that you would like to optimize. And what you would like to do, you would like to, for example, maximize this objective. So you would like to find a point in your molecular space which has the highest value of this particular objective, and then you would like to use your generative model to generate a novel molecule that actually has the highest value of your function. And what you can apply to do that is called Bayesian optimization. And basically, the idea of this framework is that you are modeling your objective function using Gaussian processes. Gaussian processes is a Bayesian method, and because it's Bayesian, it's very good at modeling predictive mean, but at the same time, is good at modeling uncertainty. And this gives you a nice balance when you are searching this space of molecules, the balance between exploration and exploitation. And to search this molecular space, you are using something that is called acquisition function. Maybe this equation is quite complicated, but the idea is really simple. This particular acquisition function is called expected improvement. And this is basically just the expected value of the indicator function that is indicated if the best current value of your objective will improve in the particular point. And finally, it has been shown that this basic formulation of Bayesian optimization doesn't work well for this molecule generation problem, but there is a nice version of, let's say, a bit more sophisticated version of this framework where you add additional constraints to this objective, and this is called constraints Bayesian optimization. And there is a really nice paper by Griffiths and Hernandez-Lobato from 2017, where they showed that basically this formulation works pretty well for this particular problem. So you're able to optimize over the molecule space, and using your generative model, you're able to generate novel molecules having optimal properties. And finally, to give you the cherry on the top, there are first commercial successes of such methods. So in July this year, the company In Silico Medicine announced that they have successfully synthesized and validated in the lab molecules that were generated using GAN reinforcement learning model. And they proved that they actually were active against a group of specific targets. So this shows that this is really nice direction of research, and that maybe in a few years from now, these methods will become mature and widely used. Thank you.